The polymerase chain reaction technique can be modified into a variety of useful modifications, including one called real-time PCR. Real-time PCR relies on all the components of a standard PCR reaction: the target DNA, the oligonucleotides, the primer, the free nucleotide triphosphates, and a Taq polymerase. The twist in the real-time PCR is this technique employs a fluorescent oligonucleotide probe, which allows researchers to monitor the progress of the reaction as it occurs in real time. The probe contains two fluorescent dyes, a reporter dye on the 5' end and a quencher dye on the 3' end. When the reporter is excited by light, it transfers its energy to the nearby quencher. This process of energy transfer, called fluorescence resonance energy transfer or FRET, it prevents the reporter dye from emitting that light. The probe participates in the PCR cycles, which begin with the heat denaturation of the DNA. The temperature is then lowered and the probe and primers anneal to the specific sequence on the DNA strands. Taq DNA polymerase synthesizes complementary DNA using the primer as a starting point. When the polymerase encounters the probe and the polymerase isn't actually blocked, the polymerase has a 5' to 3' exonuclease activity that it uses to digest the probe. At the same time, the polymerase contains to elongate the new DNA strand. The cleavage of the probe is an essential part of this reaction because it separates the reporter dye from the quencher dye. When the light excites the reporter dye can now fluoresce, and the quencher dye is too far from the reporter dye to block its emission. In the next PCR cycle, twice the number of the template DNA molecules are available for the reaction. Again, the probe molecules anneal to the templates along with the primers. As the reaction proceeds, the reporter dyes are liberated into the solution. The next fluorescence measurement pick up the fluorescence from both the PCR cycle. After each cycle, the intensity of fluorescence is measured, and the data from all the cycles are used to construct an amplification plot, as you can see. In this experiment, different numbers of starting DNA molecules from 1000 to 10 million were used in a parallel reactions. A reaction with no template DNA was also run. The time at which fluorescence increases over the background level reflects the amount of DNA in the original sample, with the earliest detectable reaction occurring in the same time with most starting materials. Real-time PCR is a quantitative assay providing the ability to compare experimental samples with known reference samples. In contrast, in standard PCR technique, the reactions are retrieved at the end of the cycle, while the reactions are no longer running efficiently due to the part of limiting reacting agents and are therefore not quantitative.